Hi, my name is Caitlin, and my data analysis project is addressing how DNA methylation differs in AM linguista honeybees and B. terrestris bumblebees when exposed to QMPs. In my experiment, I quantified DNA methylation by evaluating the relative gene expression of two DNA methyltransferase genes called DNMT1 and DNMT3. For DNMT1, I found that the difference in relative gene expression between the QMP plus honeybees and bumblebees was statistically significant. I also observed a negative gene expression for bumblebees and a positive gene expression for honeybees. For DNMT3, I found that the difference in relative gene expression for the two bee species were not significantly different. Both honeybees and bumblebees had a positive DNMT3 gene expression, which indicates that they both upregulate DNA methylation. So figure 1 highlights the relative gene expression of DNMT1 in honeybees, shown in green, and bumblebees exposed to QMPs, shown in blue. As you can see, the QMP plus honeybees had a positive gene expression, while the bumblebees had a negative gene expression. These results may suggest that honeybees may upregulate DNA methylation, specifically the DNMT1 gene, while bumblebees deregulate DNA methylation. Figure 2 highlights the relative gene expression of DNMT3 in honeybees, shown in green, and bumblebees, shown in blue, when they're exposed to QMPs. So both QMP plus honeybees and bumblebees upregulated DNA methylation due to their positive gene expression of DNMT3. Although bumblebees are observed to have a slightly higher gene expression than honeybees, this difference is not statistically significant given the p-value that's greater than 0.05. So for my data set, I use this specific figure, specifically the highlighted portions, to provide the data for my honeybees. I focused on the relative gene expression of DNMT1B and DNMT3 of QMP plus honeybees during day 1, and for each gene I calculated the averages. This figure provided the DNMT1 and DNMT3 data for my bumblebees. And as you can see from the highlighted portion, I used the shaded boxes of DNMT1 and DNMT3, which refers to the bee samples with QMP. After calculating the averages of DNMT1 and DNMT3 in my bumblebees and honeybees using the data from figures 1 and 2, I was able to create those two bar graphs displayed in the previous slides. And that allowed me to perform an unpaired t-test to determine the statistical significance of my data. To conclude, these results are important because they provide a starting point and a foundation as to how queen pheromone signaling, specifically QMPs, may affect DNA methylation in different types of bees. QMPs are released by queen bees to assert their reproductive dominance within a bee colony, and it inhibits the reproduction and fertility of worker bees. That being said, DNA methylation and histone modification is really important in regulating this type of caste specificity and managing these roles within a bee colony.